The neo-Darwinian theory of evolution, which became so important in the mid-20th century, was basically a form of Weismannism, where his theory of determinants was replaced by the idea of chromosomes and genes within chromosomes. The idea remained the same, that organisms inherit material structures from their ancestors, and that these somehow determine the shape and the instincts of the developing organism. The genes, indeed, seem to have solved the problem of material inheritance. And when Watson and Crick worked out the structure of DNA, the genetic material, in 1953, for which they got the Nobel Prize, molecular biology swung into top gear, and it looked as if all the problems of inheritance could be solved genetically through molecular biology. The idea was that the genes programmed the sequence of amino acids in proteins. The proteins may get old and decay, but the genes are replicated more or less perfectly through cell divisions. And in fact, this led to a new version of Weismann's doctrine. Instead of the germplasm and the soma, or the organism, we have DNA and proteins. And this Francis Crick called the central dogma of molecular biology. It was a molecular version of the Weismann germplasm-soma distinction. All this seemed pretty convincing, and people became very enthusiastic for the explanatory power of genes. They were supposed to be able to explain the development of form and instincts. The details were obscure, but it was assumed that they'd all be solved within 10 to 20 years. However, it didn't quite work out like that. The only way in which this gene theory could be made plausible was by ascribing to genes properties they were not actually known to have. Genes have a sequence of bases. There are 4 bases: adenine, thymine, guanine, and cytosine, which are the alphabet, which specifies in a triplet code the amino acids that are strung together to form proteins. So genes help to explain the primary structure of proteins. They don't explain the shape of your face, or the pattern on a bird's wing, or the web-spinning instincts of a spider. Those are not proteins that bear a one-to-one relationship with genes. So in order to overcome that problem, it was assumed that the genes must be a kind of program, like a computer program, that told the organism what to do. Computer programs are not simple molecules. They're intelligently designed by computer programmers. They're purposive. All these things were not supposed to be happening in organisms. But by the idea of the computer program, the genes were lent a plausibility that by themselves they didn't have. The same happened through Richard Dawkins' concept of selfish genes. The genes were endowed with the ability to shape matter, to mold form, even to be ruthless like Chicago gangsters, as Dawkins put it. They made machines, he called our own bodies, lumbering robots, constructed by the genes to propagate themselves. So selfish genes were manipulating the whole of life from the bottom up through their ruthless planning, intentions, and purposes. Again, these metaphors go way beyond anything an actual molecule could do, but the rhetoric was persuasive, not because it was mechanistic, but because it was basically crypto-vitalist. It was ascribing purpose and organizing power to molecules that they can't possibly have. Well, the enthusiasm for the gene theory of inheritance led to the Human Genome Project. Many people became extremely excited by the explanatory power of genes, and it was thought that by sequencing the entire human genome, the whole of human nature will be laid bare at the molecular level. The Human Genome Project was conceived partly because biologists have always been subject to physics envy, and the physicists had huge projects worth billions of dollars, like the Star Wars program, nuclear bombs, hydrogen bombs, space probes, the Hubble Space Telescope, giant colliders, etc. Biology was, by contrast, more like a cottage industry with small labs and small budgets. Biologists wanted a big science project that would compete with physics, be worth billions of dollars, and put biology into this sort of big science league. They needed a project that was simple enough to explain to President Reagan, and they came up with the Human Genome Project, the book of life that we'd be able to read and understand human nature. So he went for it. And the Human Genome Project was actually completed around 2000. And there was a tremendous fanfare when it was announced in the White House Rose Garden. President Clinton and Tony Blair together celebrated this triumph of biology. However, it turned out to be a terrible disappointment. People had expected there to be about 100,000 genes in the human genome. It turned out there were only about 20,000, less than half the number in a rice plant and smaller number than in a sea urchin. 99% of the genes were virtually identical with those of chimpanzees. So it didn't really explain human nature very effectively. And people who'd expected to make billions out of patenting genes found that they couldn't, and there was a collapse in the biotechnology stock market. The private genome venture headed by Craig Venter, called Celera Genomics, saw a sickening slump in its stock market valuation from over $60 a share to a few cents a share. And at least Craig Venter had a sense of humor. He said, I'm a guy who's made $1 million the hard way by working my way down from $1 billion, as he saw his holding in Celera Genomics collapse in value. By 2008, tens of thousands of genomes had been sequenced, and people were able to look at what were called genome-wide association studies to find the genes responsible for various characteristics like height or proneness to breast cancer, which were known to have a strong hereditary component. But it turned out that the genes could only explain about 10% of the difference or less. For the inheritance of schizophrenia, the genes would only explain less than 5%, whereas it was known that about 80% of these characteristics could be inherited, but the genes were not explaining what was inherited. And so this came to be known as the missing heritability problem, provoking a severe crisis in the heart of biology, because if genes weren't explaining heredity, what could? So this was a major problem for the central dogma and for the whole theory of genetic inheritance. Another problem also cropped up in the 21st century, namely a recognition that a lot of inheritance depends on epigenetics, inheritance over and above the genes. And this is basically a rebranding of the inheritance of acquired characteristics, which in the 20th century was tremendously taboo. It was often called Lamarckian inheritance. And when I was studying biology, we were told that this was a complete heresy. There's no evidence for it. There's no way that what an organism does or learns or how it grows could influence the germplasm, which was insulated from the rest of the organism and immune to influence from acquired characteristics. However, it turned out that the inheritance of acquired characteristics really does happen, as many biologists in the 20th century had claimed, but had been derided for claiming it. Some of them even committed suicide because they were attacked so violently for producing evidence for the inheritance of acquired characters. It's now mainstream, and epigenetic inheritance is one of the most exciting areas in biology. For example, One of the model organisms that biologists like using is a small nematode worm called Caenorhabditis elegans, tiny little worms, roundworms. And they've sequenced the genome, they've done the wiring diagram of the nervous system. Enormous amount is known about this worm in molecular and cellular detail. And people thought this would lead to a full understanding of its instincts and its inheritance. However, if you expose Caenorhabditis to food flavored with the chemical benzaldehyde, the worms learn that benzaldehyde is associated with food. It's a form of associative learning, and they wriggle towards benzaldehyde. If you do this for just one generation, the next generation don't respond to benzaldehyde. But if you do it for 4 generations, then for the next 50 generations, the worms respond to benzaldehyde by wriggling towards it. There's a learned response which is passed on to the offspring without modification of the genes. It's probably because of a modification of the methylation of the DNA itself, or of the proteins that bind to the DNA, the histones, or other proteins in the chromosomes. But it's not a modification of the genes, it's a modification of the expression of the genes. Genes are turned on or off in this heritable form of epigenetic inheritance. In experiments with mice, male mice were trained to be averse to a smell called acetophenone, which is a bit like cherries, by being exposed to the smell and given an electric shock. Now, when they had the electric shock, after a few times, they were terrified when they smelled acetophenone, not surprisingly. It's a classical Pavlovian reflex, a conditioned reflex. But when their sperm were used to inseminate female mice, then the children of these male mice were terrified of the smell of acetophenone, and so were their grandchildren. And this influence happened even when the sperm were passed on by artificial insemination, which couldn't give any possibility of transmission by learning or by the male mice telling the female mice somehow beware of acetophenone. And this, again, is a kind of epigenetic inheritance. And when it was published in Nature, the leading international scientific journal, the article was entitled "Inheriting the Fears of Fathers." This would have been unthinkable in the 20th century, but it's certainly thinkable now.
Find out more on advire.life.